right, good afternoon, class. You are welcome once again to our discussion on microbial nutrition. And in our last class, we basically discussed the what nutrients are and what are the various classes of microorganisms as far as nutrients and nutrition is concerned. We saw the four different types. And in this class, we'll be looking at how these nutrients are taken up by the microbial cell, the uptake of nutrients. So in this lecture, we shall be dealing with the uptake of nutrients by microorganisms and the different ways by which movement of materials take place across the plasma membrane of the microbial cell. In discussing how nutrients are transported across the plasma membrane, we will be looking at two basic processes. The first are the passive processes. Now, in the case of the passive processes, substances move across the membrane according to the concentration gradient. In other words, these nutrients, they just cross the plasma membrane from a region of higher concentration to a region of lower concentration, just like, a ball rolling down a slope. And when a ball is placed on a hill and the ball simply rolls down the slope, it does not require any energy to flow down. So in the case of these passive processes, there is no expenditure of energy. There is no expenditure of energy. It's just a flow of the nutrients from a region of higher concentration of the nutrient to a region of lower concentration of the nutrients. Now, the examples of passive movement of nutrients or transport of nutrients are simple diffusion. We'll look at them as we go along. Osmosis, as well as facilitated diffusion. Now, secondly, we have the active processes of transport. Active is like that same ball, but instead of the ball flowing down the slope, this time the ball is going up. Now, for the ball to go up, it needs energy. Something must have, pushed up the ball. So in active processes of nutrient transport, the cell must use energy. This energy are in form of ATP. It uses energy to move the substances from where the substance is of a lower concentration to where the substance is of a higher concentration. Now, a typical example of active process is group translocation. We'll look at these and others as we continue. So let us take them one after the other. Passive processes. And the first that we are looking at is passive or simple diffusion. From our knowledge of biology, rudimentary biology in secondary school, we know that diffusion is the process in which molecules move from a region of higher concentration to one of a lower concentration. Now, the rate at which this movement takes place is dependent on the size of the concentration gradient. Now, if I have this as my slope, now for the ball to move from here down to here, it is different if I have it like this. Now, if it is like this, the slope is steeper. And because the slope is steeper, so the flow is more. But if it's like this, the flow will be less. But in either case, whether like this or like this, it is passive in the sense that energy is not needed for the ball to flow down. So the rate is dependent on the size of the concentration gradient between the cell's exterior and interior. That is the places where the nutrients are flowing from and to. Now, in the case of osmosis, osmosis is the net movement of solvent molecules, and it is across a selectively permeable membrane from an area in which the solvent molecules themselves are of a higher concentration to another area where the solvent concentration is lower. Now, this continues until an equilibrium is reached. So if the molecule is moving from a region of higher to lower, this movement will continue of the solvent. This movement will continue until an equilibrium is reached, such that the concentration of the solvent is now equal at both sides. In living systems, the chief solvent is water. And the three types of solution that we can encounter are isotonic. Now, an isotonic solution is one in which the concentration is the same. So when you have an isotonic solution, the concentration of both the exterior and the interior is the same. Hypotonic, the concentration is lower than that of the cell. When the concentration is lower, hypotonic. Then hypertonic, if the concentration is higher than that of the cell. Now, in the case of facilitated diffusion, which is also an example of a simple passive movement, the rate of diffusion across the selectively permeable membrane is greatly increased by using carrier proteins. These carrier proteins are also called permeases. I will look at a diagram very soon. Now, so what we are saying here is that in addition to the fact that the substance is moving from a region of higher concentration to a region of lower concentration, in this case across the plasma membrane, there is also a carrier that facilitates this movement across the membrane. So what we are having here is a situation in which the carrier protein ferries, just how a ship ferries goods across a river. The carrier protein ferries the nutrient across the plasma membrane. Now, because the carrier aids the process of diffusion, that's the reason why we call it a facilitated diffusion. So it is diffusion, quite all right, but it is facilitated by the carrier protein. Now, take a look at this. This figure depicts facilitated diffusion. Now, what you see here is the carrier protein. Now, all these are substances, or let me call them nutrients now, that are to be transported. And this is the extracellular space outside the cell. While this is the intracellular space within the cell. Now, for the nutrients to be transported from outside inside, now what we have is this carrier protein helping to ferry or carry the nutrient across the membrane. Now, how does it do this? Now, the first thing that happens is that the carrier protein changes conformation. Now, what you can see here is that pictorially, we are describing how it opens up to pick up the nutrient. And the nutrient gets in. This is the nutrient now. And it changes conformation again to release the nutrient into the cell. So what we are seeing here is the protein or the carrier molecule, which we call the permeases, carrying the nutrients across the membrane from the region where the nutrient is of a higher concentration to a region where the nutrient is of a lower concentration. Now, in active transport is basically transport of molecules to a higher concentration. In this case, it is against the concentration gradient. And because it is against the concentration gradient, there is the utilization of metabolic energy. So, once again, this is our gradient. And if a molecule is here and has to move up, it is against the gradient. And for it to do this, energy is required. That is what happens in active transport. Now, active transport resembles facilitated diffusion in the involvement of carrier proteins activity. So there is a protein involved. But the difference is that there is the use of metabolic energy in active transport because it is against the concentration gradient. Now, one of these mechanisms that we want to consider are the ABC transporters. ABC means antibody binding cassettes. Now, ATP binding. Now, these transporters consist of two hydrophobic membrane spanning domains associated on their cytoplasmic surface, as well as two nucleotide binding domains. Now, the membrane spanning domains from a pore in the membrane and the nucleotide binding domains bind and hydrolyze ATP to drive the intake. How do I explain this? Now, it is like when we talk about hydroelectricity. When water moves in a dam, the water moves in a direction, and the dynamo moves in the opposite direction. And in the process, because of the rotation of the dynamo, it generates energy. Now, that is in hydroelectricity. Now, in this ATP transporter, it is the ATP. The ATP acts as, in fact, in real sense, there is a principle which we will consider much later on. The ATP can form this enzyme, the ATPase, and the ATPase acts like two pulleys. While one is binding the ATP, the other is helping to transport the nutrient. Now, the ATP must bind for the nutrient to be transported. The ATP binding is what makes this process requires ATP. So ATP binds, and once the ATP binds, it facilitates the transport of the nutrient. Let's see something. Now, this is the ABC transporter system. Now, right here, we can see the periplasmic binding protein. And right here, we can see the membrane spanning transporter. Now, all these are substances that are to be transported. Now, if you take a look, these substances, this particular portion, I believe you can see the arrow. This particular portion constitutes the exterior of the cell. And these substances to be transported are fewer. Their concentration is less. In fact, in this diagram, we simply used 4 of them. 4. That is 1, 2, 3, and 4. In opposition to so many that are within the cell. Can you see it? Now, here we have the ATP hydrolyzing protein. So what happens is that this periplasmic binding protein picks up the protein, or picks up whatever is to be the nutrient that is to be transported into the cell. Now here we can see the membrane. Can you see my arrow? Now this is the membrane, the cell membrane. And this is the ABC transporter complex. So when the periplasmic binding protein picks up the nutrient, once the ATP binds to the ATP hydrolyzing protein, now it is able to transport the nutrient across the membrane into the cell. So here, as we said, is the cytoplasm. Then Y here is the exterior of the cell. So the ABC transporter involves an ATP binding. And once the ATP binds to the protein, it facilitates the transport of the nutrient by the protein. Remember, we said that the ABC transporter is like facilitated diffusion. But in the case of facilitated diffusion, what we saw was that the movement of the nutrient was from a region of higher concentration to a region of lower concentration. But in this case, the movement of nutrient is from a region of lower, where the nutrient is of a lower concentration, to where the nutrient is of a higher concentration against the concentration gradient. And for that reason, the energy is needed in form of ATP. Let me repeat that. In the case of facilitated diffusion, the nutrients that is to be transported is transported along the concentration gradient, from where the nutrients are of a higher concentration to where the nutrients are of a lower concentration. So no energy is needed, but the carrier protein facilitates the process. But in the case of ABC transporting system, the carrier protein is carrying the nutrient, but it is carrying it from a region where the nutrient is of a lower concentration to a region where it is of a higher concentration. And that is where the ATP plays its role in facilitating the process. All right, bacteria also use proton gradients generated during electron transport to drive active transport. And in proton gradients, this right here, we'll talk about the chemiosmotic hypothesis. We'll look at this more closely when we start considering microbial metabolism. You see, but in proton gradients, as electrons move across the membrane, an electron is built up on one side. Now, protons are on the other side. And with the proton buildup, there come a time in which the protons will need to flow back to maintain the equilibrium. But as the protons flow back, they facilitate the movement of nutrients. We will consider that more closely later on in microbial metabolism. So what we can see here is that two things are involved in the transport. We can say it is linked. And this is what brings about, our next concept that I will show you. Now, the comparison of the three different types of transport mechanism where you have a carrier protein. Now, in uniport, you just have the substance or the nutrient that is to be transported across the membrane, and we just have just that nutrient. It is just the nutrient moving. Okay, now that is for uniport. But in the case of symport and antiport, we call them co-transport because it involves two movements. That's what I'm trying to explain. It involves two movements. Just like if you are a physics student in the pulley mechanism, where you have, two pulleys, and as one is, moving in this direction, the other is moving in another direction. Now, in symport, this is the substance to be moved. For the substance to move into the cell, another substance moves in the same direction into the cell. This is for symport. But in the case of antiport, for the substance, the nutrient, to move into the cell, another substance needs to move out of the cell. This is an antiport. And these mechanisms play out a lot in organisms generally. Now, if we take a look at group translocation, which is another form of movement across the membrane, it's an active transport. Where I want to draw your attention to in group translocation is that as the substance is transported or ferried across the membrane, the substance gets modified. Now, let us take a look at what is happening here. This is phosphoenol pyruvate plus sugar. Now, the sugar was outside the cell and is supposed to be transported into the cell. Now, what happens is that in this co-transport mechanism, pyruvate from the phosphoenol pyruvate is cleaved out such that we now have pyruvate, and the phosphoenol phosphate that is left becomes conjugated with the sugar. Now, what this implies is that outside the cell we have just sugar, but by the time the sugar has been transported into the cell, we find it as sugar phosphate. What has happened? The sugar has been chemically modified in its process of transport. So in group translocation, as the substance is being transported, it is at the same time being chemically modified. When this happens, like during prior to glycolysis, for example, we have a situation in which ATP is saved. We will consider that in microbial metabolism, because what comes into the cell is a sugar that is already phosphorylated. So, but what we are to know at this point is that in group translocation, the substance that is ferried across the membrane, in addition to the fact that it requires energy, that substance becomes chemically modified in the process. Lastly is ion uptake. It is a very unique mechanism, the uptake of iron into cells. Now, all microorganisms require iron, and this iron constitutes the cytochromes. The cytochromes are transport proteins, transport molecules. Iron uptake is made difficult by the extreme insolubility of ferric ion and its derivatives, which leaves little free iron available for transport. And that is why the process becomes active, because the microbial cell has to scavenge for the iron. Many bacteria and fungi have overcome this difficulty by a structure which they secrete. That structure are called the siderophores. By definition, siderophores are low molecular weight molecules that are able to complex with ferric ion and supply it to the microbial cell. So we have these siderophores being able to scavenge for the little available iron. And it's as a result of the insolubility of the ferric ion. So the siderophore molecules assist in scavenging for it and transporting it into the cell. Well, what we have done in this class is we have been able to look at the nutrients. Remember, we're considering microbial nutrition. We have seen the nutrients, the nutritional classes of the microorganism. And now in this class, we've been looking at the two major mechanisms for the transport of nutrients across the membrane. And these two mechanisms that we saw are the passive and the active processes. Thank you very much for your time. When we meet again in our next class, we will be looking or beginning to look at microbial metabolism. If these nutrients have been transported into the cell, how are nutrients metabolized? Till then, thank you very much and God bless you.